In the second half of this lecture, we're going to be talking about the processes of co-translational import and post-translational import, and further discuss how key proteins are trafficked to certain destinations in the cell. In this diagram, we can see the two processes of co-translational import and post-translational import drawn out diagrammatically. Up at the top of the slide, you can see what happens when an mRNA is attached to both the small and large subunits of the ribosome, and translation proceeds, producing the long, thin, purple line that you can see in the diagram, which represents the growing polypeptide. Now, in co-translational import, as the name suggests, after the ribosome attaches to the mRNA, there has to be a process that guides that ribosome and mRNA to the rough endoplasmic reticulum and allows for the import of the growing protein into the RER lumen. This is going to then allow translation to continue, but the growing protein will end up, instead of being out in the cytoplasm, it will end up in the lumen of the RER. Now remember, when we talk about co-translational import, the import is into the lumen of the RER. From that point, all of those processing steps that we discussed in the first part of this topic will occur. The processed protein will then be packaged into a transport vesicle and sent along its way to the Golgi apparatus. It will enter through the cis face of the Golgi apparatus and then travel through the Golgi for further modification. It will then be packaged again into a transport vesicle and exit out the trans face of the Golgi apparatus. From there, it's going to head to its final destination. And for co-translational import, we said there are several possible final destinations. Proteins might, for example, travel right back to the RER to live there. They may travel right back to the Golgi apparatus to live there. They may become part of the secretory vesicles or the transport vesicles themselves. Some proteins will be packaged and become part of the lysosome. Some will be sent to the plasma membrane. There are all different kinds of final destinations for co-translationally imported proteins. Now, the second part of the diagram on the right-hand side shows the differences in post-translational import. Remember, in this case, translation is not being sent directly into the RER. Instead, translation is occurring in the cytoplasm. So the protein is produced in the cytoplasm and then sent along its way to its final destination. And the specific final destinations of these proteins include the nucleus, the mitochondria, the chloroplasts, or the peroxisomes. All right, so let's begin with the process of co-translational import. Remember, ribosomes can be found free in the cytoplasm, and they can also be found bound to the rough endoplasmic reticulum. That's why we call it rough, because under the microscope, the surface of the endoplasmic reticulum looks quite rough due to the presence of these ribosomes. So for co-translational import into the ER lumen, the first thing that has to happen is the ribosomes have to become bound to the membrane of the ER. Then translation can proceed, allowing the newly formed protein to be released inside the RER lumen. It will then get processed, as we've discussed, packaged into vesicles to exit the RER, sent to the Golgi apparatus for further processing, and remember, this is often the addition or modification of carbohydrates. It will then be once again packaged into vesicles and sent to its final destination. So how exactly does the cell get this ribosome to take this translation process and bring it to the RER, and indeed allow the protein to be translated inside the RER. Some of the early details of co-translational import were described by two scientists you should be familiar with, Blobel and Sabatini, back in the early 1970s. And these scientists came up with what we now call the signal hypothesis. What the signal hypothesis tells us is that the protein itself contains what we call a molecular signal that marks it as being destined for co-translational import into the RER. We now know that any protein that needs to be co-translationally imported is going to contain what we call the ER signal sequence. In other words, within the DNA code for this protein, the very first few amino acids, about 15 to 30 of them, are not there to become part of the final protein. Instead, those 15 to 30 amino acids at the start of the protein, at the end terminus of the protein, are simply the signal or the label that says that this protein belongs inside the rough endoplasmic reticulum. Now, interestingly enough, when the ribosome contacts the mRNA and starts the process of translation, the ER signal sequence, the first part of the protein to be translated, never actually comes in contact with the endoplasmic reticulum like you might think it would. Instead, there's a small molecule in the cytoplasm in that same area that mediates the introduction of this ER signal sequence to the RER membrane. We call this the signal recognition particle, or the SRP. This molecule is made up of protein and rRNA. It's going to bind to the ER signal sequence and bring it over to the ER membrane in the area of a structure we call the translocon. What the translocon is composed of is a receptor for the SRP, a receptor for the ribosome itself, an actual pore, an opening through the membrane, and an inactive enzyme, an enzyme called signal peptidase, which again sits inactive most of the time until it's activated, and then it will cut off and remove the ER signal sequence. So let's take a look at how the cell first begins this process of co-translational import. So in the upper left-hand corner of this diagram, the light blue structure represents the ribosome, the brown ribbon represents the mRNA, and then you can see this little sort of brown tannish molecule labeled SRP. That's the signal recognition particle. So the ribosome in the cytosol is going to start translating the mRNA. And they're showing us that the very first few amino acids that get placed into the new polypeptide are colored this sort of mint green color, and then onto a purple color, and then so on and so on. So that mint green first sequence of amino acids is what we're going to call the ER signal sequence. So the very first amino acids to be placed on this new developing protein are the ER signal sequence. And this is recognized, these amino acids are recognized by the SRP. The SRP will bind to the ER signal sequence, and then if you move over to the next diagram, you'll see that it's the SRP that then carries this ribosome mRNA apparatus over to the ER membrane. The SRP binds to its own receptor in the membrane, in that structure we call the translocon. It also guides the ribosome in to bind to its receptor. And then in the next segment of the diagram, we see that GTP is going to get hydrolyzed. And that's because we need something to happen to the translocon at this point. We need the pore portion of the translocon to open, and that requires energy. And the energy comes in the form of GTP. GTP gets hydrolyzed, the pore opens, and the signal sequence is guided into that pore. You'll notice that they show it sort of looped in the region of the pore, and that's because the signal sequence is actually going to bind to the wall of the pore. Now, during this time, those first three drawings at the top of the diagram, during that time, we don't want the ribosome to keep translating this protein. We want to wait until the signal sequence is in place and the pore is open. So temporarily, at least, the SRP is capable of not only guiding everything into place, but also stopping translation temporarily until everything's in place and translation can begin once again. Now, if you move to the bottom half of the diagram, over on the left-hand side, you'll see that once everything is in place and translation has restarted, the SRP gets released. It's done its job. It's guided the ribosome and the mRNA where it needs to go in contact with the translocon. The pore is open and translation can continue. In the diagram, they're showing us this by showing that the purple part of this polypeptide is getting longer and longer and longer as the ribosome travels along the mRNA. Eventually, the entire protein is translated and released into the lumen of the RER. At that point, the pore is going to close, the ribosome will be released, it will separate and release the mRNA. So, again, co-translational import involves several key steps. The first thing that has to happen is that the SRP has to take the ribosome down to the membrane of the RER and get it in contact with the translocon. The translocon has several pieces to it, but its most important job is to act as a channel for the translocation of the protein. In other words, the translocon needs to allow the protein to be transported into the RER as it's being translated. GTP is hydrolyzed to provide the energy, and the ER signal sequence is what tells the SRP that this protein needs to be imported into the RER. Once the pore opens, remember, the SRP has done its job and it will be released. And again, what we can see in this diagram is that the SRP is really critical in getting all the pieces together, in getting the ribosome and the mRNA in contact with their parts of the translocon. The SRP has a receptor, the ribosome has a receptor, and once GTP gets hydrolyzed, that pore will open, the signal sequence will bind within the pore, translation will restart, and that protein will be translated while it's being imported into the lumen of the ER, co-translationally. Now, as the polypeptide gets translated into the ER, the signal peptidase, the portion of the translocon that has enzymatic activity, is going to be activated, and it will remove the ER signal sequence. In other words, the ER signal sequence has done its job by getting the newly formed polypeptide into the pore while it's being translated. Once that happens, the signal sequence is no longer needed. So by the time the protein is completely translated, the signal sequence can be removed. At that point, once the polypeptide is complete, it will be released from the translocon and the pore will close. And again, this is demonstrated on the bottom part of this diagram, which shows us that on the lower left-hand side, the signal sequence is bound inside the pore, translation restarts, the purple strand gets longer and longer, feeding down into the pore, but then the signal peptidase, shown in gold, is going to act to cut off the signal sequence. This is going to release the new polypeptide into the lumen of the RER. So any protein that's destined to be co-translationally imported into the RER is going to have 15 to 30 amino acids at its N-terminus that act as a signal. Now, other molecules within the cell are used as signals to take proteins to different locations. In other words, there are other signal sequences. There's certain signal sequences that mark a protein as belonging in a lysosome. There are other sequences that mark a protein as belonging in a peroxisome, or a protein that needs to head right back to the RER. There are different signal sequences for different destinations. Now, once a protein gets co-translationally imported into the RER lumen, all the things that we talked about in the first part of this topic are going to occur. So this includes, remember, first off, splicing and any cleavage that needs to happen. Secondly, chemical modification. Now, we talked about some of the ways that chemicals can be added or removed from this processing protein. But one of the things I want to stress here is the process of glycosylation. Most of the proteins, actually, that get targeted to the ER are going to become glycoproteins. They're going to have carbohydrate added to them. Now, there is some terminology you should be familiar with here. These carbohydrate side chains that get added, if they're bound to a nitrogen on the protein, then we're going to call this N-linked glycosylation. If the carbohydrate side chain ends up being bound to an oxygen in the protein, we're going to call that O-linked glycosylation. So these newly formed glycoproteins will continue on with the processing that we discussed last time, and will eventually get packed into transport vesicles so that they can travel on to the next part of their destination journey, which is travel to the Golgi apparatus. Remember, these proteins are going to enter through the cis phase, travel through the Golgi, and exit through the trans phase. While they're in the Golgi apparatus, they will be further modified. And remember, in the Golgi, that's primarily either glycosylation or modification of glycosylated proteins. So we're either going to add more carbohydrate or we're going to modify the carbohydrate that has already been added. All of these carbohydrate modifications are there to allow proteins to reach further destinations in the cell. After all the chemical modifying is done, now it's time to fold the protein. And we'll spend a couple minutes talking about this process of folding because it's so very important to the final function of the folded protein. Once a protein is in the ER lumen and it's time for folding to take place, this process is actually facilitated by a large group of proteins called chaperone proteins. Now, there are lots and lots of different kinds of chaperone proteins, but the most abundant type of chaperone is actually called HSP70, or heat shock protein 70. Now, this protein originally got its name because it was originally described in connection to its appearance after cells were stressed with heat. In other words, when the heat was applied in the laboratory to these cells, the amount of heat shock protein 70 would go up. We now often refer to HSP70 as BIP, which stands for binding protein. Either one of those terms is fine for us. So we're going to find HSP70 in large amounts near the translocon, near to where proteins are coming in to the RER, on the cytoplasm side of the membrane. The BIP molecule is going to bind to particular regions of that new protein, hydrophobic regions, in order to first prevent that new protein from simply folding in some random way. So in other words, BIP's first job is to keep the protein unfolded until it can be folded in a very particular step-by-step manner. While BIP is attached, other chaperone proteins are going to come in. They're going to attach to particular regions on the protein, and they're going to start to guide the folding process. So folding has to happen in a very systematic way, step by step by step. If you allowed protein folding to occur spontaneously in the cell, every time the protein folded, it would fold a little differently. Remember, proteins are very large, so there's lots of different ways any one protein could fold. Because protein has to fold in a very systematic way, step by step by step, it requires energy. And that energy comes from ATP hydrolysis. What the energy is used for specifically is to allow these chaperone proteins to bind, guide a fold, then unbind and rebind somewhere else. Guide a fold, and then unbind and rebind somewhere else. So folding is an energy demanding process. This picture represents the folding protein that we call HSP70 or BIP and where it sits within the cell. So in the picture you can see the membrane designated by that yellow strip region. You can see outside of the cell up at the top of the slide, inside of the cell down at the bottom. The ribosome is depicted in purple, the mRNA is a red strand. We can see a new polypeptide forming inside the channel of the ribosome, and we can see it coming through the pore of the translocon and into the cytoplasm. The red structure that you can see inside the pore would represent the signal that got this protein to the ER to begin with. Now BIP is represented in sort of a light blue color. It's floating in the cytoplasm, and you'll notice it's floating right around the area of the translocon. It's there because it needs to meet that newly forming protein as soon as possible, bind to it, and prevent it from randomly folding. So the chaperone proteins are going to bind to particular regions on this new polypeptide, and they're going to help the protein fold in one particular way. All of the folding that occurs involves interactions between the R groups on each amino acid in the polypeptide. So remember, it's the R group chemistry that matters in a protein. All the amino acids share certain structure. It's the R groups that are different from amino acid to amino acid. So depending on their charge and their polarity, and whether or not they have partial charges within them, if they're hydrophobic, all of those things matter. That's going to allow the chaperone proteins to bring certain amino acid groups together to form hydrogen bonds, certain groups to come together to form van der Waals interactions, and there are other types of molecular attractions that can form that we didn't really talk about in this course. For example, disulfide bonds are important between the amino acid cysteine and other amino acids that contain sulfur. So the molecular chaperone proteins bind and guide the folding of each new polypeptide into its mature protein form. Now some proteins are not going to fold normally, despite the action of the chaperone proteins. Remember, biological systems are not perfect, and mistakes get made. So when a protein starts to fold abnormally, when it misses a particular fold or it makes a fold it shouldn't have made, processes can be set into motion to fix this. First of all, the BIP will bind again to try to hold the protein in a particular conformation until the mistakes can be repaired. Or if the mistakes are too far gone, the protein can be destroyed. So there are two particular types of responses that we need to be familiar with that the cell makes when a protein misfolds. One is called the unfolded protein response, or the UPR, and the other one is called the ER-associated degradation response, or the ERAD response. Let's talk about each one of these separately. When a protein starts to fold abnormally, it triggers a response. The first of those is the unfolded protein response. There are particular sensors in the ER membrane that can detect a misfold, and we won't go into those specifically, but what happens when a misfold is detected is we're going to get another one of those temporary translation stop messages. So at that point, whatever part of the protein still needs to be translated and brought into the ER, that's going to be put on hold. Translation is going to be put on hold temporarily. One of the things this allows for is an enhanced translation of more chaperone proteins. So other ribosomes will be busy for the process of translating other chaperone proteins and some what we call degradation type proteins. So degradation proteins are just enzymes and complex bodies of multiple enzymes called proteasomes, whose job it is to take apart and destroy a misfolded protein. Generally, the cell wants to identify a misfolded protein so that it can unfold it and refold it the correct way.